So now let us just start, and I'll continue with other things as we go on. So the first bacteria I'm going to discuss here is Pseudomonas aeruginosa. Now, when you come or when you hear Pseudomonas aeruginosa, the first thing, aero means what? Aerobic, isn't it? It's a strict aerobe. It is, it needs oxygen. Absolutely, it needs oxygen to survive. Means what? Where will it go to? Which organ will it go to get the highest oxygen? Can anyone tell me?
The lungs, sir.
Exactly. So it causes majorly lung infections. And other key points, I'll come to all the descriptions, but just when you hear, because the first thing which, whenever you hear a bacteria, something should come to your mind, which you're able to connect it, because, if nothing comes to your mind and everything you're by hearting, then it will become, you don't know what you have learned. Okay. So two things which should come to your mind. One is it is causing lung infections. Second is it causes hospital-based infection. It's called what? Anyone of you know that what is it called? Hospital-based infections. Any specific name? If you don't know, it's fine. It's called a nosocomial infections. Okay, nosocomial infections. So these are the key two things which should Pseudomonas aeruginosa, hospital. Okay, and it's that it's one of the dangerous things which can happen to a patient, isn't it? He's admitted in the hospital, for example, for some kind of disease, and is getting another disease from the hospital. See, this bacteria can even survive in your disinfectants. Okay, we'll come to sterilization and disinfection and how to kill this bacteria. That's a different topic. We'll discuss it in Pseudomonas aeruginosa. So today we'll be discussing the topic under these headings. Okay, one is gram staining, the oxygen requirements of this bacteria, as I've told you, how to culture this bacteria and identify and diagnose the disease, and the diseases which it causes. Okay, these are the key areas we'll be discussing today. So this is basically the bacteria. You can see there the gram negative, okay, bacilli, that is rod-shaped bacteria there. So that's it, gram negative bacilli. Okay, you can see there, all of you. So basically oxygen requirements. So they are, as I've told you, they are strict aerobes. Okay, and they are oxidase positive. Means they produce an enzyme called oxidase. Now oxidase, we'll come to oxidase produced in our body, human body. Okay, when we'll be discussing general pathology, where our body produces oxidase enzymes to kill other bacteria like NADPH oxidase, peroxidase, and all those. So similarly, this bacteria also produces oxidase to kill other bacteria. But the key feature here is they're oxidase positive. Now you're going to get questions. Gram negative bacilli, which is strict aerobe, oxidase positive, and causing this disease. What is identify the organism? Okay, or you can also get this image. Okay, this image there. Okay, this image, and they can give you this description, oxidase positive and all, and they ask you to identify the organism. Okay, so now people say that how can I identify from this particular image? Because any bacteria can be this. Now see, they fool you. The examiners fool the students by giving such images. Now this image has nothing to do with identification unless you're a microbiologist. Basically what the key points are, they will give you some key points. Oxidase positive, strict aerobe, gram negative bacilli, causing nosocomial infection. So these are the key words which you should identify, not from the image. Okay, now there are some questions where you get image-based questions, directly you understand it from the image, but you can also get this kind of questions is what I mean. Now oxidase positivity, that is they produce oxidase enzymes. Okay, as I've told you just now. Now one more oxidase positive organism to know is Brucella. Okay, just to connect it. Okay, oxidase positive organisms. Now like you should be able to identify between these two. Okay, oxidase positive, two organisms you studied. One is Pseudomonas aeruginosa, just now what we're discussing, and Brucella. We come to Brucella today. Okay, another bacteria which we'll be discussing today is Brucella. Okay. Now in culture, now I'm going into a little bit of basics now because you're just starting your preparation, isn't it? So basically, hope you have gone through the basic bacteriology lectures and studied about culture media. And why do you want to culture the bacteria? Usually you get from a sample, you get a very little bacteria to identify it. So you grow it more, you multiply it. So for that, you culture it. And certain bacteria require specific growth requirements. Okay, they need specific food to grow. For example, if you just take our case, some people love biryani and they eat biryani, they become more fat. Similarly, okay, or some people like burgers, like they need some selective requirement. So the selective requirement of this bacteria, that is Pseudomonas aeruginosa, is citramide agar. Now this can itself be a question. Okay, citramide agar is a selective media for Pseudomonas aeruginosa, they can ask you. Another thing is, when they are grown in the selective media, okay, they will multiply and they start producing pigments. So this will help us to identify the organism that this bacteria is growing. That is, they produce 3 kinds of pigments. One is pyocyanin, other is fluorosin, third is pyorubin. So just know, okay, one more thing, it's good to write down the notes. Okay, quickly write down the notes, rough notes at least, and quickly review after a lecture so that you retains whatever you have just listened right now. So pyocyanin, blue. Okay, fluorosin, okay, its other name for fluorosin is pyoverdin, and pyorubin. Rubin means red. So blue, green, red pigments. So this is the picture here where Pseudomonas aeruginosa growth is indicated by producing this pigment. Pyocyanin, blue-green pigment, pyorubin, and fluorosin is also there. Here it's showing that nutrient agar, basically, it can much better grow in cetrimide agar. Okay, agar in which cetrimide is added. That's the way to remember it. So that's the culture medias, and that's what the pigments they produce. So far we studied the gram staining, the oxygen requirements, it's an oxidase positive. Culture media, very, very, very important, cetrimide agar. Okay, and yes, the pigments they produce. Now again, let me go a little slow. Asking you, this is where they can ask you strict tear-offs, gram-negative bacilli producing oxidase positive, producing blue-green pigments in cetrimide agar. Identify the organism. They can give you this image, any image they can give you. So that's a very clinical level questions you can get. Now the disease. So which bacteria are we discussing about? You should not lose the touch with. Pseudomonas aeruginosa. Exactly. So what are the diseases? Now, as I've told you, the most common agent producing hospital-based infection. Okay, this is a question in itself. Most common agent causing nosocomial or hospital-based infections. That is this. Now, another key point, most common or most serious organism producing infection in burns. We'll learn in detail about burns in general surgery. Okay, so what you'll learn this bacteria again there. So most serious infections in burns patient is caused by Pseudomonas aeruginosa. Now, basically, just remember it like that. Patient is burnt. Okay, the patient is taken to the hospital. So then hospital infection, nosocomial infection, isn't it? Another is most common gram-negative. We studied this is gram-negative organism. So most common gram-negative organism producing cystic fibrosis. Now, cystic fibrosis is a disease of lungs. Okay, we studied when we begin. We studied that this organism aeruginosa likes air, likes oxygen, so goes to lungs. So any big problems of lungs, this can infect. So cystic fibrosis means, you'll learn this in general medicine. Basically, cystic fibrosis means cyst in the lungs which get fibrosed. Fibrosis means what? Fibers get deposited, becomes hard. So a cyst there in the lungs which gets fibrosed. Just remember it like this for the time being. Okay. And another disease it can cause is Shanghai fever. Okay, it can cause Shanghai fever. So these are the diseases it causes. Now here, I just want to add in nosocomial infections, I just want to add a definition of nosocomial infection. We studied Pseudomonas aeruginosa that it causes nosocomial infection. So what is the definition of nosocomial infection? So just remember this picture, okay? Pseudomonas aeruginosa hospital, okay? Just for you to remember it. So what is the definition of nosocomial infection? They've asked you, okay? Not Viva, a question, which of the following correctly depicts nosocomial infection? So there are 2 ways to define a nosocomial infection. Okay, the infection which happens or which appears 48 hours or more after hospital admission. Okay, the patient is admitted to hospital. After that, if is get infected within 48 hours, it is a nosocomial infection. That's one definition. 48 hours after. Okay, now another, or within 30 days after discharge. Now patient has no problem, he was in the hospital, no issues, but got discharged. After discharge, he got infected by 30 days. Within 30 days. Now what is the reason for that? Incubation period. Okay, this bacteria has got a very long incubation period because it gets inside the patient's body. It can even go up to 30 days to create an infection or symptoms. Okay, so these are the 2 definitions of nosocomial infections. Okay, and just know that this picture is important, that if you see this. Okay, that's the definition. Now the next we studied burns and cystic fibrosis. So Shanghai fever basically it's a very mild kind of illness. Okay, self-limiting febrile illness resembling typhoid. Okay, so Salmonella typhi You might have come across the lectures on that, producing typhoid fever. So similar kind of infections, okay, is being caused by Pseudomonas aeruginosa. So this is basically the disease. Okay, so if you have taken down notes, just quickly look into the notes. So we studied gram staining. We studied that they're strict aerobes like Brucella. Sorry, they're strict. They're oxidase positive like Brucella. I'm sorry. Then yes, cetrimide agar. Don't forget this keyword. They produce blue-green pigments. So even if you don't remember the name of the pigment, it's still fine. But please note the most important key point is nosocomial infection. What is its definition? And second is Burns patient, cystic fibrosis, and yes, we can get Shanghai fever is caused by which organism? It's Pseudomonas aeruginosa. So all of you understood?
Yes, sir.
Okay. Others, I'm expecting you also to respond. Okay. Now, yes, fine. Now coming to the next disease, Yersinia pestis. Can you make out what is being shown there in the image? Anyone? It's called a black death. Okay, black. Okay, death. Okay, that's death is coming. Black death. Okay, and the other name for plague. So Yersinia pestis, like I told you, if you hear Pseudomonas aeruginosa, you should think of hospital and lungs. Like similarly, Yersinia pestis, you should think of basically plague, black death, very dangerous problem. So here we will discuss. These are the topics we're going to discuss. Okay, other names of it. Okay, then other name of this organism, then the diseases it causes, definitely plague, how to identify it, and how it is being transmitted. Okay, you can see there is small cute rat there. That's okay. Now, okay, so Yersinia pestis, black death, and rats. Okay, so other name, it's good to know, it's called plaque bacilli. So it's bacilli. Okay, gram negative bacilli, same thing. Like we discussed about Pseudomonas aeruginosa, gram negative bacilli, plaque bacilli. Now, what is the disease it causes? As I've told you, it's causing the plague. Okay, it causes plague. Okay, now plague was a very, very dangerous infection which was earlier there before, but now, yeah, almost not non-existent. Okay, okay. Now, next, okay, plague, a little bit more details you want to know. One is, as I've told you, it's also known as black death. Okay, the other name for plague is black death. Now, I'm not going into too much of details of this, but yes, few things I just want you to note. Okay, that is different types of plague. Okay, different types of plague has been asked as a question. Okay, so there are three major basic types of plague. Okay, one is bubonic. I'll tell you why it's called a bubonic. Second is septicemic. Third is pneumonic. Now, basically, again, these are again organisms which can go into the lungs. Okay, because they are again loving oxygen, not as much as your Pseudomonas aeruginosa. Bubonic means basically what is bubonic? And basically, its infection of this bacilli initially begins by infecting the lymph nodes. So you get a painful lymph node. Okay, and that painful lymph node is known as buboes. Buboes, the name. So that's why it's called a bubonic type, and it's the most common. Now, this is a question you can expect. Most common type of plague, okay, is bubonic plague. Okay, or the most common infection caused by, or most common type of manifestation caused by plague is bubonic type or painful lymph nodes. Now, next is septicemic. Now, it gets into the blood, and just septicemia, it just spreads up into the blood. That's called septicemic. So from lymph node, it goes into the blood. Okay. The third thing, possibility is pneumonic. It goes into the lungs. Pneumonia, isn't it? Lungs. So now here, this is the most infectious type. It can spread. You know, it goes into the lungs, the patient starts coughing and it can spread. Understand it, isn't it? So very, very important 2 points. The most common is bubonic. Most common type of plague is bubonic, and most infectious type is pneumonic. Okay. Now, another way they can ask you this question here is, all of the following are types of plague except bubonic, septicemic, pneumonic, and some other terms. Okay. They can simply add some vague terms, convalescent type, that type, this type. No, only 3 types are there: bubonic, septicemic, and pneumonic. Okay. So these are basically the types, okay, of the disease. So we studied one is the other name, what it causes, plague. Again, different types of plague. Now, the patient got plague, so you have to identify and diagnose it. So you have different methods to identify it. Okay, one is you culture it and you stain it. Okay, and if that stain is being caught, you can say that this is the organism. Okay, so yeah, you can see out there, that's the staining. I'll show you. If you're using GMSA stain, okay, this is GMSA red stain. There are different stains to learn, but yes, just know GMSA red stain. If you're using, you'll see that bipolar safety pin appearance. If you see this more intense staining, bipolar, like a safety pin. So that's the bipolar safety pin appearance on when you stain it with GMSA red. Another stain you can use it, Weissen stain. Okay, so once again, a quick thing I just want to tell you here. Okay, I'm sorry. You have to identify it, you stain it. Okay, plaque bacilli, you stain it with GMSA or Weissen stain, you get bipolar safety pin appearance. Okay, now, how is the transmission? Understandably, plaque, you know, it's a parasite of rodents. Rodents means rats, okay, and it's transmitted to man through the flea bite. So the flea goes and bites a rat, comes and bites human, you get it. Okay, so this is the most important key points I just want to tell you here. Yersinia pestis, so the key term which should come to your mind, or key picture should come to your mind, is rat and black death, which is plague. 3 types: bubonic, septicemic, and pneumonic. Bubonic is the most common, pneumonic is the most infectious. Okay, now you identify this bacilli by the bipolar safety pin appearance when you stain it with GMSA Weissen stain. Okay, finally, it's a parasite of rodents. Now, I just want to teach you a method of learning here, which is called developing photographic memory. Now, one way people learn is how, you know, they by heart everything. Okay, one they start by heart everything. Tomorrow they take it, or they try solving some questions. They say that, sorry, I forgot everything. Now, that is very normal. Another way is you're learning here the key points. Just now see how I explained. It took me 10 minutes or 15 minutes to explain it in total. But now when I review it, I quickly catch the key points only. Now this key point is what you have to learn initially. Okay, you have to read everything. But when it comes to learning, today, for example, you should just learn Yersinia pestis means plague, and which is black death. 3 types: bubonic, septicemic, pneumonic. Most common is bubonic, most infectious is this thing. Okay, now how to identify both bipolar safety pin appearance. Forget about the stains and all right now. Okay, and remember the rat. So if this summary is there in your mind, this picture is there in your mind, or a picture of your notes is there in your mind, when you revise it multiple times, other things also get set. So the first picture will be your key areas. Slowly by slowly, this picture goes into your mind. Okay, and you start learning everything. In that way, go and sit in the examination hall. It becomes super easy for you to recollect from what you have learned. Instead, you're by hearting everything, you forget everything. If you're by hearting only the key points, you remember that. Okay, so let me go to the next bacteria, which is Haemophilus influenza, another organism which is associated with pneumonia and lungs. Okay, so whatever we have studied so far, it's related to the lungs. Okay, now Haemophilus influenza. Now you know that influenza is one of the very notorious infection you find out in you see with most of the time. Okay, it can be bacterial, but at times it can be influenza virus also. So this is bacterial Haemophilus influenza. Okay, so here what we're going to discuss is again the other name of it. Okay, what are the growth? Now Haemophilus influenza, unlike other bacteria, it has got some specific growth requirements. Okay, other bacteria also have some culture medias and all, but very specific. Haemophilus influenza, the key thing which should come to your mind is growth requirements. Okay, growth requirements. Haemophilus influenza means the growth requirements. Identification tests are there for you to identify it. Again, it's related to growth requirements. Okay, now the other name of this bacilli is Pfeffer bacilli. Now the person who found out his name is related to Pfeffer. So it's known as, again, it's a bacilli, Pfeffer bacilli. Now growth requirements, it's a most important thing I just want to teach you. Factor V and factor X. Some people say it's factor 5 and factor 10. Now it can confuse you with your coagulation factors, which is factor 10, factor 5 there. So it's a different thing. This is a different thing. Okay, this is again related to human body, but that's coagulation. This is growth factors. Okay, now factor 5, factor 10 is needed if you want to grow Haemophilus influenza, or you can say that Haemophilus influenza get into the human body. Okay, get into the human body, and what it does is it takes this factor 5 and factor 10 from the human body and it grows. Okay, one way. Another way I'll tell you. Now, factor 5, the other name is the NAD. Okay, what is NAD? Have you learned biochemistry? NAD is
nicotinamide
adenine dinucleotide, or you'll be more familiar with NADH, okay, which produces ATP, or from which you get ATP. So you get a question, how many ATPs are produced from NADH, like that. So yeah, the same NAD, which is also known as factor V. Okay, now this NAD is derived from a vitamin called niacin, vitamin B3. Okay, so you'll study all this in biochemistry, but just want you to connect this at that time when you perform. So the best way to learn for, am I audible? Am I audible?
Yes, sir. Yes, sir.
Fine, right? Okay, the best way to learn for NEET MDS is to connect things. Okay, you learn 20 subjects to connect them together. Okay, wherever possible, it becomes super easy. So like that, so they are derived from NAD is factor 5, other name for it, and it's derived from niacin. Okay, and one more thing, we are talking about which bacteria? Haemophilus influenza. So Haemophilus influenza require factor V and factor X, whereas para-influenza, another bacteria which is related, requires only factor 5. You can get a question like this. Okay, you can either get a question, para-influenza growth requirement is factor 5, factor 10, both A and B. Answer is factor 5. Or if it is Haemophilus influenza, factor 5, factor 10, yes, both A and B. Okay, so they are derived from niacin. Another thing is, yeah, this will study in biochemistry. NADH produces this, energy and all. We'll study that. Now, factor 10, the other name for factor 10 is hemin. So it's getting from hemoglobin. Okay, hemin is a part of hemoglobin. We'll study that later. So when it comes to Haemophilus influenza, the important thing is growth requirements, factor V and factor X. What's the time? Okay, after 45 minutes to 1 hour, we'll just take a break and come back. Okay. Now, identification test, a very important test. You culture them, you get what is known as satellitism. Okay, now it's again related to your growth factors. Just know that. That is the Staph aureus. We studied Staph aureus in our lecture. Staphylococcus aureus. They produce these factor 5 and factor 10. So this bacteria, Haemophilus influenza, depends on this particular Staphylococcus aureus to grow. So that's called satellite. Okay, getting it from somewhere. Okay, satellite, you know what they do? They get the range or radiation for us from other places. So it's kind of, that kind of thing. So that's satellitism. You can get this, you can see this picture there. Okay, where you have Staphylococcus aureus, which is growing here. Okay, here in this gray area, more grayish area, you could call it as. Okay, this area. Okay, and you have Haemophilus influenza growing around it because it's getting the growth factor. So area with very visible growth is this. Okay, the white dots, whereas here you see that there's very little growth. Okay, so it's blood agar is going in. Okay, and along with the Staph aureus, so it's called satellitism. So they can simply ask you, they can either give you this picture that ask you identify the bacteria. Maybe they'll mark you that Staph aureus and all, or the Smith's practice, definitely. So they can ask you this way, or they can simply ask you satellitism is shown by which bacteria, Haemophilus influenza. So a quick review. H. influenza, the Pfeffer bacilli, the other name. Key point is factor 5, factor 10. Okay, factor 5 and factor 10 for its growth. Whereas para-influenza, only factor 5. Identification, satellitism. You learned influenza. You revise it continuously, revise it. Every other points will go. Okay, and please, when you start your preparation, don't worry about your pending topics. Don't worry about anything. You're just taking your first step of your preparation. You start worrying, you'll stop your preparation. So just keep going. We have 3 rounds to cover of all these subjects in different methods, in different ways. Okay, first will be the very basics understanding, then we'll understand the basics plus a little bit of complicated. So right now, just cover up. Okay, now the third and the last bacteria for this session I'm going to discuss is Haemophilus ducreyi. Again, Haemophilus influenza, para-influenza. Now ducreyi. Now here I'm going to discuss with you, one is the lesion and other is identification. Not everything, just lesions, a very typical feature of ducreyi. Ducreyi causes sexually transmitted disease. Okay, sexually transmitted disease. Now, yes, the lesion is known as chancroid. Have you heard of anything called chancre?
Yes, sir, syphilis.
Yes, similar kind of lesion. You can see this, those genitals producing the ulcerations. Okay, now what is the difference is, here it's painful ulcerations. So H. ducreyi produces painful ulcerations of the genitals. Very, very important. Do you know, can you tell me, Dr. Carol, was the syphilis chancre was painful or painless?
It's painless, sir.
Painless, yes. Syphilis, so that's one difference. So chancroid is painful, chancre is painless. Okay, chancre is painless. Now, another thing is identify the organism. Now, it's not a very important topic, just for you to learn it. You have one is known as ITOS test. Okay, ITOS test, ITO. Okay, and next is you can stain it. Okay, so that's how you can identify it. Okay, and yes, here you are getting satellitism. Remember, don't forget satellitism here. Now, an ITOS test, you get a school of fish appearance. Now, it's like fishes are going for school. That's all. This fish is going to school. So these are the H2 Okay, school of fish appearance. Very, very, well-mannered line. That's why it's called school of fish. Okay. Another thing it can do is you can go for a staining where it produces bipolar staining, like we just saw where. Okay. Where did we saw bipolar staining?
Yersinia pestis.
Exactly this. Okay. Bipolar staining, safety pin appearance. But here it's just on the either side it stains. There is no safety pin appearance. On either end it is staining. Okay. Now, bipolar staining. Second thing is railroad track appearance. You can just remember this picture itself. Railroad track. So, school of fish, railroad track. Now, H. ducreyi, what should come to your mind is your chancroid and the school of fish. Okay, if that is there, fine. Okay. So, what we'll do is we'll have a short break. We have a few more bacteria to discuss. One is Bordetella, Brucella, we studied oxidase positive, Mycobacterium. Now, we're not discussing Mycobacterium, we're discussing atypical Mycobacterium, because Mycobacterium detailed lecture is there. We'll try to finish it as much as possible. Spirochetes, okay, all the spirochetes, and specifically Treponema we'll be discussing, and we'll also discuss Leptospira. So whatever possible we'll discuss today, and then we will have one more session. We'll have 2 sessions enough initially, and the other sessions we'll postpone it to next week. Okay, so take a 5 minutes break, come back in 5 minutes. We'll just have one more session as much as possible. Okay, thank you.